Welcome to the second part of Cell, the Unit of Life. Plasma membrane. Plasma membrane is an absolute requirement for all living organisms as it is responsible for the relationship of a cell with the outside world. The detailed structure of the membrane was studied only after the invention of electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane enabled the scientists to deduce the possible structure of the plasma membrane. The chemical studies done specially on the human red blood cells enabled scientists to deduce the possible structure of plasma membrane. On the basis of these studies, scientists found that cell membrane is composed of lipids that are arranged in a bilayer. These lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tail towards the inner side. The polar ends interact with the water and are hydrophilic. This ensures that the non-polar tail of saturated hydrocarbons or hydrophobic tail is protected from the aqueous environment. Later, biochemical investigation clearly revealed that the cell membranes also process proteins and carbohydrates. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% of proteins and 40% of lipids. Depending on the ease of extraction, membrane proteins can be classified as integral or peripheral. The peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. The integral proteins which run through the bilipid layer are known as tunnel proteins or transmembrane proteins. These proteins cannot be removed easily, and their removal requires crude methods like treatment of detergents. Thus, the membrane has been described as protein icebergs floating in a sea of phospholipids. An improved model of the structure of cell structure was given by Singer and Nicholson in 1972, and it is widely accepted as a fluid mosaic model. According to this, the quasi-fluid nature of the lipids enables lateral movement of proteins within the overall bilipid layer. The ability to move within the membrane is measured as its fluidity. Functions. The fluid nature of the membrane is important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Plasma membrane allows the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. The passage of substances across cell membrane occurs by various methods such as passive transport and active transport. Passive transport. Many molecules can move across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to lower concentration. Water may also move across the plasma membrane from higher to lower concentration. The movement of water by diffusion through membrane is called osmosis. As the polar molecules cannot pass through the non-polar bilipid layer, they require a carrier protein of membrane to facilitate their transport across the membrane. Active transport. It is an uphill movement of materials across membrane where the solute particles move against a concentration gradient from the lower to higher concentration. Such a transport requires energy, which is obtained from the adenosine triphosphate ATP. Thus, it is an energy-dependent process. For example, sodium-potassium pumps seen in animal cells. Cell walls. The cells of bacteria, fungi, algae, and plants have additional non-living rigid structure called the cell wall that surrounds the plasma membrane. The composition of cell wall varies in different groups: fungal cell wall, algal cell wall, plant cell wall. Let's talk about the fungal cell wall. The fungal cell wall is generally composed of chitin, a polymer of N-acetyl glucosamyl NAG units. Algal cell wall. The algal cell wall is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Plant cell wall. The plant cell wall is chiefly composed of insoluble polysaccharides like cellulose. Certain other compounds such as hemicellulose, pectin, and proteins are also present in the cell wall. The cell wall of plants consists of two regions: primary wall and secondary wall. Primary wall. A young plant cell forms a single layer of wall material. This layer is known as the primary cell wall. The primary cell wall is thin, elastic, and capable of expansion in the growing cell. It grows by the addition of more wall material within the existing one. Meristematic and parenchymatous cell have primary cell wall only. Secondary wall. In mature cell, more layers of wall material are added to the internal to the primary wall, which gradually diminishes with maturity. These layers are called secondary cell wall. Addition of secondary cell wall brings about thickening of the cell wall. Thickening of cell wall occurs particularly in cell that forms the harder woody parts of plants such as lignified and suberized cell wall, which do not allow water. Middle lamella. Adjacent cells in the plant tissues are held together by thin, sticky, amorphous layer of cementing material. This layer is called middle lamella. Middle lamella is chiefly made up of calcium and magnesium pectate. In ripening fruits, the pectate compounds solubilize to a jelly-like material, making the fruit soft. The cell wall and middle lamella may be traversed by plasmodesmata, which connects cytoplasm of neighboring plant cells. Functions of cell wall. The cell wall serves many functions. It maintains shape of the cells, it protects the cells from mechanical injury, it wards off the attack of pathogens like viruses, bacteria, fungi, etc. It allows materials to pass in and out of the cell. It helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Now let's talk about pits. The cell wall is not uniform in thickness throughout, because at certain places secondary cell wall is laid down. Such unthickened areas are called pits. Pits of the adjacent cells generally lie opposite to each other and form pit pairs. Pits are of two types: simple pit and bordered pit. Simple pit. In the simple pit cavity is uniform in diameter. Bordered pit is flask shaped as in tracheids. Plasmodesmata. It forms from the living component on the cell. It forms the living component in the dead wall. A number of plasmodesmata or cytoplasmic strands are present in pit through which the cytoplasm of one cell is contact with other. These are lined by plasma membrane and contains 5 tubule called desmotubule. Endoplasmic reticulum plays a role in origin of plasmodesmata. These forms the symplastic system between the two cells. Now let's talk about the endomembrane system. The interior of eukaryotic cell is composed of many membrane-bound organelles. Each of the membrane-bound organelles has a distinct structure and function, but some of these organelles function in a coordinated manner and constitute an endomembrane system. The organelles included in this system are endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. The functions of mitochondria, chloroplast and peroxisomes are not coordinated with the above mentioned organelles, hence these are not considered as part of endomembrane system. Let's talk about endoplasmic reticulum first. Electron microscopic studies of eukaryotic cells reveal the presence of network of reticulum of tiny tubular structures scattered around in cytoplasm. Those are called as endoplasmic reticulum. Ultrastructure. The endoplasmic reticulum is composed of the following three kinds of structures. They are cisternae, tubules and vesicles. Cisternae. The cisternae are long, flattened, parallel, sac-like interconnected structures. These are found in the cells which are actively involved in protein synthesis. The cisternae usually occur in those cells which have synthetic roles. Example: cells of pancreas and brains. These are usually associated with large subunit 60S of ribosomes. Tubules. The tubules are branched and unbranched structures forming the reticular system along with the cisternae and vesicles. They are free of ribosomes and are common in cells involved with lipid and sterol synthesis. Vesicles. The vesicles are oval, membrane-bound vacuolar structures. These are also free of ribosomes. They are abundant in pancreatic cells and these are the only endoplasmic reticular structures found in spermatocytes. Endoplasmic reticulum divides the intracellular space into two distinct compartments: luminal compartment and extraluminal compartment. Luminal compartment. It is the internal space enclosed by the endoplasmic reticular membrane. Extraluminal compartment. It is the space present outside the ER in the cytoplasm. Types. On the basis of presence of absence of ribosomes on the surface of endoplasmic reticulum, it is divided into two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. The endoplasmic reticulum, which is free of ribosomes, is known as SCR. When it is observed under the electron microscope, it appears as smooth tubular structures. The muscle cells are also rich in smooth type of endoplasmic reticulum, which is known as sarcoplasmic reticulum. Functions. It is specialized in synthesis of lipids and steroids, detoxification of drugs associated with muscle contraction by the release and uptake of calcium ions. Synthetic products of RER pass through to Golgi complex through a smooth endoplasmic reticulum.